Hi, I'm Keith Seifert. I'm with Agriculture and Agri-Food Canada, and I'm here today with my professor, the man who introduced me to mycology 41 years ago.
My God.
How do these things happen?
I don't know.
Bryce Kendrick. And Bryce was a professor at the University of Waterloo for about 30 years. And Bryce, why don't you tell the camera where we are right now?
We are now sitting in a Garry Oak meadow at the top of John Dean Provincial Park in British Columbia on Vancouver Island. And this is a particularly interesting place because I came up here a couple of years ago and counted hundreds and hundreds of Lycanomphalia mushrooms, which depend on algae that grow on the surface. So the meadow is surrounded with trees which are mostly either Garry Oaks or what we call on the west coast, what do we call them on the west coast?
Bob.
Yeah, Bob and Andrew. Yeah, it's an ericaceous tree.
You're thinking of the arbutus.
Of the arbutus menziesii, that's right. This is where the memory sometimes does not serve.
Oh, wow.
Not immediately, anyway.
So we'll explore your memory then.
Yeah, what's left of it.
Or what's left of it, yeah. So people can tell now from your accent that you're not from these parts. So can you tell us a little bit about your background and where you came from and how you got interested in biology?
Yes, well, I was born in Liverpool, which was then a very dirty industrial city. And I grew up having to walk a mile or so to get to the library. But we had good parks, and my father was very keen on getting us out into nature. And I started collecting leaves, and he bought me a leaf book. And I started to look at this leaf book, and I found a leaf that was not in the book. So that caused me to explore a bit more widely, and I discovered that it was the leaf of a tree that was not native to Britain, but which came from the Carolinian forests of North America. And that is and what is the name of the tree?
I have no idea. Pawpaw?
Yeah. So once I had found out that this ericaceous tree, I didn't even know what the family Ericaceae meant at the time, but I knew that it was a very different tree from anything local. And that sparked my interest in the wider field of biology. I also lived on the edge of the ocean, and we used to go to the beach at Formby and Freshfield and go swimming in the Mersey, which was a fairly polluted river at the time. And it was exciting because there were the wrecks of many ships that had been torpedoed there during the war. And I have lots of stories about the Second World War that don't belong here, that have nothing to do with biology or mycology, but still. And I found in the sea where I was swimming these little tiny semi-transparent, almost transparent creatures swimming around. And I put some in a jar and took them home. And next morning they had vanished. So this piqued my curiosity, and I had to find out what they were. They were sea gooseberries. They were members of the phylum Ctenophora. And they are some of the most beautiful objects in the world because they have rows of flagella that march down the sides of the jelly and that beat in a meta-synchronous rhythm.
Okay.
And when the light shines on them, they refract the light and send out little rainbows. So they're incredibly beautiful things. So that again made me very interested. And we took courses in marine biology. We had a field course in the Isle of Man, and there was a lab there owned by the University of Liverpool, where I went with my high school group, and we looked at some of the things that were growing there. Again, a lot of members of the various different phyla that one expects in the marine situation, which you never see on land, dead man's fingers and things like that. So yeah, I was very clued into biology from a pretty early age.
So your father was, what did your father do?
He worked for Meccano, and he was an income tax clerk.
Not Meccano, the toy company?
Yes, the toy company. Meccano, Dinky Toys, Hornby electric trains. And I, of course, was well supplied with all of those things. So if I had been going to be an engineer, this would have set me on the right track for that. But I was interested in them, but it didn't take. The biology took, and marine biology especially. So when I went to university, I tended to focus rather on that aspect of things.
So you went to university
at the University of Liverpool because we had just got rid of a conservative government at the end of the war, and the new Labour government decided that anybody who had the ability to take advantage of university education should get it. So I was given free tuition, etc., to go to the University of Liverpool. And admittedly, I had to run my sixth form year twice because I was too young to go to university the first time I did it. But when I was 17, I went. And when I was 24, I finished my PhD. But there's a lot in between.
24, that's pretty young.
Yeah, well, it just went straight through. It took me 7 years. 4 years undergraduate and 3 years in graduate school.
So who were your mentors? Like who were the people who were supervising your theses?
Well, I had a very interesting young plant physiologist who used to take us out to interesting locations not far from Liverpool, like salt flats, and at one point we went up on a field course to Malham in Yorkshire and explored the limestone pavements there. So there are a lot of very unusual plants growing there, which really excited me. And I think from then on it was the botanical side that interested me, not the zoological, although I'd started off being more interested in animals, marine animals and so on, but then I got into the plant side. And it seemed to me that the people in the botany department were more accessible than the people in the zoology department. The zoology department had long corridors and big doors, and they were always closed. The botany department had short corridors and lower doors, and they were often open, so you were able to talk to the profs. And I had a very inspiring lady prof in first year, and her name was Marjorie Knight, and she was an algologist. She discovered the life cycle of one of the brown algae.
Oh, okay.
And she was an amazing character. She had lost a leg somewhere, so she went around on crutches, and she smoked like a chimney. But she coughed when she breathed in the chalk dust from the blackboard. But she fascinated us, and I can remember her coming up behind me during a lab and say, "Mr. Kendrick, I'm sure you can do much better than that." So she was both stimulating and invigorating and somehow authoritarian. She pushed you. And she was great. And she became Dame Marjorie Knight, and she retired to the Isle of Man and ran a pottery there for many years. So she was a fascinating woman. I had another woman who was a prof, an algology prof too. And the year my mother died, she took pity on me and took me on a field course that she was running a research problem in the Isle of Man again at Port Erin, which is where the marine biology courses had been. And we had a diver who went down to the bottom of the bay and charted the positions of various algae and so on. And I was doing various things in the boat, like triangulating our position and so on. And she was very interesting. Her name was Elsie.
Elsie.
Oh,
you knew her by first name.
I knew her by first name, yeah. Although she was a somewhat stiff individual, she had a very attractive young daughter that I was totally enamored of, who was along on a trip, so that was a plus. But anyway, so she was an algologist, and I was kind of drawn towards the idea of working with her for a graduate degree. And then I looked at what had happened to the various students that had taken honours with her. And they all got third-class honours, which disqualified them from doing graduate work, essentially. So I said, I can't throw myself in that particular pyre. And about that time, we got a new faculty member from Australia, an Australian professor who came by way of Queen Mary College, and he was a mycologist, and he had looked at the decay of the leaves of the she oak, Casuarina, in Australia, which is where he came from, of course. And when I asked him if I could do honours with him, he said yes. And so I got an immediate exposure to mycology, because he took us out into the woods, the pine woods, and showed us various mushrooms and things. And I wasn't totally into mushrooms, but he seemed to me to be a very impressive academic, because he had come from a long way away, and he was the new chairman of the department, and he had some clout. So when I finished my honours and got a 2-1, nobody at Liverpool had ever got a 1 by that time, although somebody did later on. So and I said, would he take me on as a grad student? And he did. And it was almost disastrous, because he was a terrible supervisor. He was overbearing and authoritarian, and he was never there, because he was into Universities Council for Higher Education overseas, which meant in the Commonwealth. So anyway, he was rarely in. So he fortunately hired a young guy by the name of Dennis Parkinson, who came from a women's college in London to teach at Liverpool. And he arrived just in time to save me from the flames, because I had been given a project by this guy, and he said, look, when we isolate fungi from the soil, most of the ones we get are light colored. But when we look at fungi in the soil, most of the hyphae we see are darkly colored. Can you find out what the connection is there? So I started isolating these dark hyphae, and they're all dead, of course. So eventually Dennis came along. Dennis Parkinson said, well, he says, let's look at the different layers and see what happens from one to the next. And so I started dividing the organic layer of the forest floor. This was a Scots pine forest in Cheshire. And I divided it up into layers, and fortunately the Scandinavians had done all this before me, so I was able to pick up the names, the L, the F, the F1, the F2, the H. And although their Swedish name or the Danish name was humuskiknet, I just called it the humus layer. And the F layer was the formultningskiknet or something, and I just named that the fermentation layer. So this was much simpler for me to deal with. And I then started making observations and setting up experiments, and these things worked out. So I was able eventually to establish a kind of a series of fungal zones that progressed through the different layers of the forest. And in so doing, I found some new fungi. And that was the big key. I found 2 new hyphomycetes. One of them we called Helicoma, and it's been kicked around since then, now has its own genus. So
that's the same one we found in your front yard here.
We found it in my front yard, growing on a completely different pine tree. On a Californian pine, yes.
And the other was, of course, Thysanophyra.
No, that was another new one, but the new Sympodiella, a new genus. And it's still a new genus, still a good genus, and it has more species now. So that was very exciting. So I got to go down to London. I met Martin Ellis and talked to him about these various things, and he said, oh yeah, that's a new species of Helicoma. Well, he and I were both wrong. It wasn't a Helicoma at all. And it was many years before it was finally put in its own genus, and that's a whole different story. But this goes into the way in which you gradually develop a deeper understanding of these things the more you work with them.
Well, and something can be true at the time you do it, and then 30 years from now, there's a new truth to be discovered, and so the whole field just kind of evolves?
Well, at that point, we were just looking at helicospores. They just curved around. Okay, if it's a helicospore and it's growing on a single conidiophore, it's probably a helicoma. But the fact was, helicoma was a thing that produced its spores by growing sympodially and producing them on teeth and things like that, which this didn't. This was a single spore sitting on the end of a hypha.
Yeah.
So it didn't fit into helicoma ultimately, and now it's got its own name, which is Slimacomyces.
Slimacomyces.
Yeah.
So,
and it's still monotypic, I think.
I think maybe there's another species now. I think there might be one from China there.
Hey,
but
probably.
So and then at some point you graduated and you moved to Canada. So what was behind the move to Canada, and how was that?
Well, when I finished my PhD and got it through the Viva, I was looking for a job, and they said to me, if you come in a year or so, we can probably get you a job at Kew in the CMI and that kind of thing, because they were still in an expansion phase, I guess. However, there were two other jobs that came up at the same time. One was at the University College of the West Indies in Kingston, Jamaica, and the other one was a postdoc in Canada with Stan Hughes in Ottawa. And I didn't think I was quite ready to go out and teach in the tropics, because I knew nothing about them. I'd never been there. And I thought the idea of working in an English-speaking country with somebody who was already a very famous person in the field that I was interested in was more encouraging. So I chose the Canadian route, and I never got to graduate because I was on the ship coming to Canada when I should have been wearing my mortarboard and robes. I've never worn them.
on the ship, right? So that it was kind of a one-way trip.
Yeah, it was a one-way
those
trip.
Days, right?
I never went back.
Yeah.
And after I'd been here a year, the Canadian government offered me a job in the group that I was working in, and which has changed names several times since then.
Yeah. Well, and so then you went to Waterloo, and so I was a student at Waterloo. And one thing that was striking about that department to me was that it was basically all expats of, from different countries, the United States, from the UK. So, what was it like, making that transition from a I guess really a postdoc and government scientist into a teacher?
Yes, well, I had been with the government for 5 years at this point. I was pretty fed up with the bureaucracy. I don't like bureaucracy, and I don't like red tape and things. So the idea of a university job began to be more attractive all the time. And at that point, the person who had been the chairman at the University of Liverpool moved to Waterloo and started to build a new department. And the year I went, the faculty went up from 7 to 12. And he had hired people from all over the place. As you say, he got people from the States, he got people from England. And it was a growing department, and NSERC was doing quite well. I liked the idea of, not as I didn't know it at the time, becoming a small businessman. But that's basically what you are as a faculty member. You have to go out and get money to keep your students. Well, at least I had to do this. I had to get grant money. And on the one hand, that was stressful, but on the other hand, it was a chance to expand, to explore new ideas in new areas. So if you look at my CV, you'll find we wandered off into various corners of what the hyphomycetes might and might not be doing. Well, growing in streams and growing in ponds.
You seem to let your students to follow wherever their noses took them. So that you had students working on mycorrhizae, on aquatic hyphomycetes, aquatic hyphomycetes, coelomycetes.
That's true. I thought if they had a bent for something, they should do it. And as long as it was mycological, and as long as it was to do with what they used to call imperfect fungi, I was happy with it. So we never did, well, actually I did have a student who worked on mushrooms, but that was because he absolutely insisted on it.
Yeah.
And I said, you'll never get a job in mushrooms, but you get a job in molds. And you know what he finished working up? He finished working with aerobiology, with molds and buildings. And he's still doing that.
That's Dewey.
Umpty years later.
Yeah.
So that was, but generally, yeah, I like to let them do what they wanted to do.
So there's one thing I wanted to talk to you about, and I don't think we've ever discussed this really, and that's the Kananaskis conferences. So you organized these two conferences in the 1970s that the second one in particular ended up being quite a seminal meeting in the field. So I'm curious how you came up with just the thought to have these conferences and how you managed to organize them. And
it's difficult to remember all the details at this point, but I'd been to a botanical congress and I thought, wow, the potential of a meeting of this kind, but restricted to people in the one discipline, was very attractive to me. So I thought, if we can get people like Subramanian from India and people like Chris Perzinski from England and Martin Ellis and so on together to talk about this particular group of fungi was kind of exciting. And in fact, when they got together the first time, even at the first Kananaskis, a lot of sparks were struck. I think you really have to get people together and kind of lock them up in the same room to make them interact at that kind of level.
So good sparks, not
yes,
conflicts.
good. Well, yes,
both.
mostly good sparks. Yeah, and we got people together who were looking with electron microscopes at these fungi and looking at the fine structure, and people who were looking at the development, and people who were looking at the ecology, and you kind of stir them all together and they come up with new things.
So were they all self-funded? Did they come on their own dime, or did you have to find money?
I had to find money to bring several of them, and I applied for a grant, and of course it was inadequate to run the whole thing, but we had a very cheap venue in the forestry lab at Kananaskis, and it was a beautiful place to be, and we had a couple of great field trips into the Rockies during the course. So well, it wasn't a course, it was a confab.
Confab.
Yes.
But one of the things that's very striking about both those books is the transcriptions of the discussions,
Yes.
which as a student, and I know a lot of my colleagues who also were influenced by those books, we probably read the discussions more than we read the papers, because that was where the development of the ideas was articulated. So was there a precedent for that? I think that Ron Peterson had done a book on Basidiomycetes that also had transcribed
something like that.
discussions,
But I felt very strongly that the discussions could be the nub of the whole thing, because you know, a presentation, a paper, is it possibly a chapter in a book, but the literature's full of that stuff.
Yeah.
What it's not full of is the minutiae of the interactions of the people who write the papers. And frequently, what led to the papers, it never gets into the paper. So it's best to let these people stew among themselves and then try to transcribe as much as possible. I really tried very hard. It took me months,
I bet,
to do all those transcriptions. But yes, I found that they were an important part of the book. The two books.
Yeah, the two books.
And the National Museum paid to publish them.
Yeah. So the program was seminar presentations followed by discussions, or
yes.
was it more organic?
I
guess
think
the discussions would be organic.
Yes, there were seminars. People talked about what they wanted to, and then the rest of us piled in and said, well, but, if, and when, and so on, and kind of tried to draw more out of them or push them into corners. And this is when people are often the most revealing.
Sure. And you know, what's striking about some of those discussions is that we're still talking about some of those things today. And
yes.
One thing that comes to mind is the term Lirioconidium and chlamydospore, which were kind of discussed at that conference, and there was really no resolution. Other than that, maybe we shouldn't use those terms because they're not well defined, but people still use them, and or not. You know, reviewers say, oh, you shouldn't use that term, but it captures that discussion. So we don't that kind of discussion, we don't really see that so much anymore. So another thing that was very striking about your career after that time was that you became this kind of publishing entrepreneur, and that's very interesting too. And you mentioned that being a professor in a way had that entrepreneurial expectation, but even nowadays we have people who's kind of set up their own little publishing company around their labs or broader, and I think you were kind of an inspiration for some of that. So what led you to go in that direction?
Well, John Semple and I did a textbook for a second year course, which covered the whole of, he dealt with the plants and I dealt with the algae and the fungi. And we put it together as a kind of a ring-bound thing. But then I decided that I wanted something more specific to the fungi for the third and fourth year courses. And it led to my writing a series of chapters to try and cover everything I possibly could, which has expanded since then. But at the time, I thought it was fairly comprehensive. And I borrowed $5,000 from the bank and went to a guy up in Owen Sound who was a kind of a small publisher. And he produced the first edition of The Fifth Kingdom, which is a horrible thing in the sense because they stapled it together with giant staples. And I remember somebody getting in touch with me saying, the first thing I did was rip those staples out.
Oh, really? And what did they do? Put it in a binder?
I don't. They must have bound it some other way. But that was an inspiration because the guy encouraged me and I got my $5,000 back eventually. So I then went on and did a second edition, and that got picked up by a guy in the States. I'm sorry I wasn't able to stay in Canada, but the Canadian publishers were not keen on laying out money, and at that point I didn't want to go borrowing anymore. So they put out the money for the second and the third editions, and when it came to the fourth edition, I had a new US publisher because my original US guy had retired. And these people wanted a much more gussied up version. So they said, we'll put some color in, we'll allow you to expand, add extra chapters if you want, expand chapters. And so I was catapulted into that at the age of about 82. I didn't really totally want to do it, but I didn't not want to do it. I didn't want it not to be done, So I got myself into the gear again. So the first edition came out in 1985. And the last edition, the fourth edition, came out in 2011. So that's quite a long stretch there. Yeah. Particularly since the third edition came out in 2001. So we've got
10 years.
17 years.
Yeah.
17 years between the two editions. It was really too out of date. And I found that when I was teaching this course at Bamfield this year, that some of the students, they were supposed to get my fourth edition, but that's quite expensive. It's 500 pages, etc. And it's expensive to ship. So they had taken advantage of an offer from the publishers to buy the third edition for a lot less money. But there's a lot of stuff that's not in the third edition, which we now need to know. All the molecular stuff is new from between the third and the fourth edition. And lots of stuff about fungicides and things like the kinds of ways in which we exploit fungi have broadened out. There's stuff about, fungi made from fungal mycelia and car parts.
Oh, yeah.
And so on, so that the uses of fungi have multiplied. And things like the strobilurins, which I had never anticipated because I was wandering through the forests up here and finding these little tiny white mushrooms growing on last year's Douglas fir cones. Had no idea. I knew they were called strobilurus.
Yeah.
Which is a good name for them because they grow on cones. But I had no idea at that point that they were responsible for this huge outbreak of fungicides, which affected almost all groups of fungi. You say they don't deal with
doesn't work with
fusarium
very well.
That's too bad. So the banana disease is not
unfortunately
cured.
Yeah.
No.
It's not registered to use with Fusarium.
No, interesting that.
So last night I was looking at the diplomas that you and awards that you have hung up on your home lab wall,
office wall,
and saw your Distinguished Mycologist Award from the Mycological Society of America from 1995. And I was there when you got that award, and I remember you made a very short speech, and it was basically that I owe all of this to my students, and so I'd like you to talk about that relationship that you've had with students and continue to have now.
Well, I think what I discovered this summer at Bamfield was that the thing I enjoy doing most is teaching undergraduates. This beats almost any other occupation I can think of by a country mile, because their minds have reached a level where they can grasp some of the concepts, and they're also possibly capable of becoming enthused by something, by having ideas grab a hold of them. You don't see that in small children. I used to think I'd like teaching small children. It's not true. So these undergraduates aged between 21 and 26 were just the perfect group for me to work with. They came from several different universities and they had different backgrounds, but they uniformly became very keen on the fungi. They call themselves myco-nerds now. and they're still corresponding with me months after the course is over and sending me pictures of fungi and things like that. And I go back far enough. I think that must have been happening all through the times I was teaching at Waterloo, because the occasional undergraduate would become a graduate, and then others came from somewhere else. But the idea of having someone who is looking at something different from what you're interested in, but is now able to communicate to you something exciting about that area that you didn't know. And so I remember, for example, Mark Brundrat, who is now in West Australia and has written a wonderful book about West Australian orchids, working on the arrangements of mycorrhizal fungi in the spring flora in the woods near where he lived. And it was absolutely amazing to see the timing of the entry of the endomycorrhizal fungi into the roots of those plants as it related to the leafing out and flowering of those plants. Sometimes they were totally out of phase.
Yeah.
I was stunned by that. I couldn't understand what it was all about. But the thing with mycorrhizal fungi is I've been reading a few papers lately, and all they do is confuse me, because one of my ex-students, who is now a prof at UBC in Okanagan, did some very good papers on endomycorrhizal fungi, and he found that they are as often harmful to the plant as they are beneficial to it. Well, that just blew me out of the water, because I had been brought up on this concept that the mycorrhiza was always a beneficial, a mutualistic symbiosis, and it isn't. So where do we go from there? I read these papers. I can't make sense of them. I want to sit down with him and talk about it, but I don't know when that's going to happen. Maybe next fall.
Well,
Yeah, so the students all through. See, you and Gracia and one or two other people are working on this hyphomycetes on dung thing.
Yeah.
I was very keen on that as an undergraduate exercise, but there's been some amazing fungi discovered on dung. Just gorgeous things.
Yeah, I've grown out of that phase now, but
Yeah, you don't.
I remember it fondly.
I used it again at the Bamfield field course, and we got some magnificent things. It strangely enough went straight from Pylobolus into the mushroom phase. There was very little in the way of ascomycetes in between. But in our years with the horse dung, we got used to get a long sequence. So you're not looking at dung anymore.
Not very often. we have deer in the yard, and I sometimes pick it up in the spring, but I used to see Polaria, and that's about it.
Yeah.
I think they have a boring diet in the winter.
Yeah, yeah. Well, this is one of the problems, isn't it, of getting dung that's got the right ingredients.
Yeah.
Yeah. So yes, I credit graduate students with enriching my life tremendously, because they opened up areas that, as far as I'm concerned, were locked, and they exposed them to me, and just as I hope I was exposing certain things to them on the way in.
Yeah.
Then they take off on their own like racehorses.
Well, they should. Well,
And
yeah,
they did. I mean, look at John Kay.
Yeah.
He's a fellow of the Royal Society now.
Well, I think that, you know, you exemplify a lot of what this kind of professorial academic lifetime relationship is like for a lot of people in science, you know, and you've managed to keep in touch with a lot of the students that you've had, and they've wanted to keep in touch with you. You know, it's a permanent kind of relationship, right?
Seems to me.
It's not a transient thing.
Should go on forever.
Yeah. Well, we'll see how it goes.
Well, we have little get-togethers of grad students who can actually find the time to come and celebrate my birthday at off months, because who wants to come in December?
Yeah.
So they came at different times of the year, and we had a slightly wet weekend where we explored all kinds of ideas and old friendships. And that was very good. I'd like to expand that, but our house isn't very big.
Yeah. So you retired at a little bit
60.
ahead of schedule and moved out here to Victoria, and it's like, in a way, you kind of moved back to where you started. You know, living on in a coastal environment, and
Yes.
back. I think your plan was to explore marine organisms again, and
I
so
did.
you've done that, but you've also unexpectedly done other things. And so if you want to talk for a few minutes about the environmental aspect of your retirement.
Okay, well, I knew we used as a family to go out east one year and out west the next year, and we alternated these things, and we finally decided after several alternations that we wanted to finish up on the west coast. So in in the 80s, I started looking. So I came to a conference at UBC, and I came over to Victoria, and I got a real estate agent who very smartly had advertised in the University of Waterloo newsletter. Interested in retirement or investment property, call me any time of day or night. So she took me around, and I found a place that I liked the look of. So I was taking video of everything, and I took it back and showed Laurie, and Laurie says, no, it's too big. She says, I want something on the waterfront. So next year I said, you come with me. So she came out, and we did the routine again. We found the place we're in, which was a bit of a wreck. But anyway, my main aim in coming out here was to get into a new biome, a different biome from the one I was living in. Now, it's not as rich in trees as the one in the east, because we don't have any Carolinian forest. It was all pretty well, everything was scraped off here. But we do have a lot of interesting plants and animals, birds that were not present in the east. It's absolutely wonderful to be surrounded, as we are, where I sit on the edge of the ocean with. There's birds every day of the year. There's hummingbirds. We have Anna's hummingbirds coming and exploring everything, and we have juncos, dark-eyed juncos, big bunch of them around, and siskins and crossbills, and all kinds of strange things come through. And we have 2 or 3 eagles that sit at the top of a tree next to us and talk. They don't nest there, but they stay there for hours at a time. And so I enjoyed that, but I'm not a real birder. I'm a kind of a semi-keen birder. I'm interested in them, but I don't spend a lot of time looking for LBJs. And I don't know where everyone is in the bird book, like the people in South Africa did. But I then started looking at the marine organisms again, and I compiled a CD with 650 species in it over a period of about 5 years after I got out here. And I still go back and look at that, because it's got all kinds of pictures of things I'm not seeing as often anymore. And that makes me wonder, because I've been back to Heron Island several times since my original visit in the early 70s. And each time I've gone back, it seems that there is somewhat less biodiversity than there was the last time I came. And this bothers me because I think it's happening around here. Around this peninsula, I can see there are algae that I'm not seeing anymore. There are invertebrates that I'm not seeing anymore, and I just wonder whether this has to do with temperature change, climate change, whether it's got to do with man's incursions on the ocean, the pollution, or what. It just seems there's a slight withering away, and the same thing seems to have happened with birds and mammals. They've all declined quite strongly over the last decade or so. And this bothers me. So I thought I was coming out to paradise here, and that I would relax and just look at the oceans and the forests. And the forests here are wonderful, of course. We've got some great trees. But in fact, all I do is worry about them. And then that got me into the concept of going out and trying to get rid of invasive plants. And I've now spent a good chunk of the last 3 years cutting broom. And I started off cutting broom that was 15 feet high, and now I'm down to cutting broom that's maybe 3 feet high, but we're getting rid of it. And you can't do it everywhere because it grows all along the highways. And when the last time I flew back from San Francisco, I saw a band of yellow all the way up the coast. You can't get rid of it. But we can get rid of it in a park or a reserve, and we can keep it that way and help some of the rare plants that grow there to flourish. And that's what I'm engaged in now, as much as anything else. And I'm gathering material for the 5th edition of the book.
Okay. Well,
have you done me?
I've done you. Thanks, Bryce. That was great. And I should say happy birthday to you.
Oh, thank you.
Someone's having
coming
his
up soon.
85th birthday in 2 days from now. So. And I wanted to do this interview up here at this spot because we've been up here several times together, and we've been out around and about this peninsula, and I've watched you cutting the broom out. So
yeah.
you remain an inspiration. So thank you for everything.